Okay, so today we're going to talk about biotechnology heuristics. So we have spent the whole class talking and teaching you guys how to design and implement and make genetically modified organisms. And now I guess maybe this isn't appropriate on the very last day, the final last lecture, to spend just only an hour talking about some of the consequences of that and some of the ethical, I guess, conundrums and contemplations that one should consider before you do all those things or before you implement these things in the field. So let's start. So a first question is let's just ask, oh, it always does that to me. Let's just ask why. Like, why would people be worried? Why would some people be worried about making genetically modified organisms? And is some of that worry valid? What are the big concerns? Well, one of the things people worry about are mutations. And we've talked a lot about how to make mutations specifically. Usually when we're making mutations, we know the desired outcome. But one of the things that we worry about is if we implement some kind of a genetically modified organism strategy and we put that into the field, into the wild, what's going to happen when evolution starts happening on those genes, selection starts happening on those genes? They're going to change over time. And a big question is, what is the effect? What's going to be the effect of that change over time? So imagine we have a scenario where we created a mosquito and we used CRISPR-Cas9 and we used a gene drive to change some receptor so that it can't be recognized by a particular virus. And in theory, imagine we did this, and in theory, this is now going to create a transgenic mosquito which can no longer spread that particular virus, and yay, we've saved humanity. What happens if that receptor, after we release it into the wild, what happens if that particular allele mutates and changes in a way that nobody predicted, and it mutates in a way that actually does the opposite? It's not that it can't be recognized now, it's actually like super recognized. It's even, the affinity is even higher for the virus, and so the virus is now even able even more to invade particular mosquito cells, and it makes that mosquito a super vector. Now we're spreading a super vector mosquito, which is transmitting this virus at a higher frequency if that rare scenario happens. And the question is that possible? Okay. And I think it is. It is possible for that scenario to happen. So we need to be very, very careful about when we make a genetically modified organism, we have to be cautious about putting that and implementing that into the field directly. This is just one example. A second thing that people worry about is what's called horizontal gene transfer. Okay, and we worry about this a lot in the context of antibiotic resistance. So in all of the microbial strategies we have been taught, that I have conveyed to you, you have probably now noticed that in all these transgenic strategies for making genetically modified microbes, there's always a selectable marker. There's always a selectable cassette. And most of the time in microbes, in bacteria, we're using an antibiotic resistance cassette. Okay? And one of the things that we worry about is if this antibiotic resistance cassette is also a resistance cassette for an antibiotic that might be therapeutic for humans that we might use in the hospital, we worry that if we're releasing bacteria into the wild and they have these plasmids with these resistance cassettes, what are plasmids famous for? Like when we originally defined plasmids, we talked about one of the very unique things about plasmids is that they're almost designed and evolved to share information, to share that genetic information. So when we start releasing plasmids and even just the genes themselves, the antibiotic resistance cassettes inside of bacteria, when we start pumping those genes, those alleles into the wild, we really worry that that bacteria might spit out that plasmid. And there might be another bacteria in that environment which could then pick up that plasmid, okay? And this bacteria might be really bad. It might be a plant pathogen, it might be a human pathogen or an animal pathogen, and now we've created a very, very bad scenario where this bacteria that we often have to try to control for, or we often have to treat for, now has antibiotic resistance to a particular antibiotic that we might be using. So we really worry about this phenomenon is called horizontal gene transfer. This is when an organism has a particular gene and that gene somehow is mobilized and gets transferred to a completely different species. Okay, that's horizontal gene transfer. And this happens all the time. This is not uncommon. We know how genes move. We know that if a gene is on a plasmid, that can be shared. We know if a gene is close to transposons or mobile elements which jump around, those genes can be shared and transferred. And so we really worry about scenarios where we're putting genes into an environment that might not stay in that environment. They might get transferred, and that could cause serious consequences downstream. A third thing that we worry about is we worry about the health of the ecological systems. We worry about ecological collapse. And I am not a community ecologist. I'm not an expert in ecology. I'm not going to claim to be. But I do know that in ecology, there are sort of what everybody knows are like the cornerstone species, where if you have a bunch of species diversity, there might be a very, very key cornerstone species which is really important for that ecological environment. It might do something very important in the soil. Maybe it's fixing nitrogen or doing something if it's a microbe. Maybe if it's an animal or a plant. Maybe it's degrading biomass in a way that supports a whole bunch of insects in the soil. There are these cornerstone species which sort of facilitate the niche in which all these other species live in, okay? And if something happens to that cornerstone species, you can have a cascading sort of collapse of an ecosystem. And a lot of these ecosystems can be perturbed by like invading organisms. If all of a sudden this sort of evolved ecological system, which has been sitting there for maybe 1 million years, if all of a sudden a new transgenic creature is now like invading this ecological niche, that could really cause a disruption. And we don't always know what the mechanism or what the downstream effect is going to be to the release of that genetically modified organism. So this is something that we worry about, and we have to think about it. And a fourth thing that we worry about a lot, which I've talked about before, especially with relation to insecticides, is off-target effects. And I also talked about this in CRISPR-Cas9 system, where when you're using a nuclease that has a guided homing target, it's not always guaranteed that that target is going to match that one thing. It might match a couple things. And so you might get editing in a couple different places in the genome. And there's a question of what is the consequence of those off-target effects. In the insecticide community, the famous example of this is if you treat land or say like turf grass with neonicotinoids, those insecticides, the residues of those insecticides stick around and they get on bees, and then the bees bring them back to their what do you call them? their hives. They bring them back to their hives, and then all of a sudden everybody in their hive is now like being killed, murdered with these neonicotinoids. So these are off-target effects when we're trying to do something, but really we only have a minimal effect on what we're trying to do, but we have a gigantic effect on something else, which we weren't trying to do. That's an off-target effect. So these are all four things which are really a significant consideration with respect to GMOs. So mutations, horizontal gene transfers, ecological collapse or ecological impact, and off-target effects. So back to the original question, I really do think that the original question was, are there things that we should be worried about? Should we be cautious about making genetically modified organisms and just willy-nilly releasing them into the wild? And the answer is yes. There are some really serious things that can happen if we're not cautious about that. Okay, but before I outline these heuristics, I do want to just say, though, that now we are not in the boundary of science anymore. When we start talking about sort of morality or ethics, a lot of people won't like this, but I'm going to say that's not the realm of science. Like, it's not so clear-cut. Science is supposed to be about facts and what you can observe. What can you clearly observe? And what can you conclude from those clear observations, okay? And where can you find converging data sets that sort of tell you the truth of a system? That's what science is about. Ethics and like how we should approach things, that's not the realm of science. So I just want to be clear that everybody's going to have their own systems of philosophy and ethics and heuristics. And so these are sort of my personal principles, which I just think it's good for me to share them now during this course. And I'll just give citation. A lot of this I have pulled from a philosopher who's written quite a lot of books, a modern sort of, he's like a mathematical philosopher. His name is Nassim Taleb. I like his books a lot, okay? I previously was not so much of an environmentalist. I was very gung-ho about we should be engineering our environment. And then I read Nassim Taleb's books, and he has some really convincing arguments about how to think about carefully doing things. And so I just want to give credit that a lot of the things that I'm going to be talking about are with respect or pulled from Nassim Taleb's books. And I should even be cautious myself, because Nassim Taleb is famous for making fun of people who like get his books wrong. So I'm probably going to get a whole bunch of it wrong, and that'd be awesome if he made fun of me someday. But anyway, I just wanted to give credit where it was. His two books, he's got 5 books that I've read, but his 2 books that are most important for sort of coming up with this lecture are Anti-Fragile and The Black Swan. So here's Anti-Fragile. You should definitely read it. It's a really good book. And here is The Black Swan. The Black Swan is not quite as interesting as Anti-Fragile, but you have to sort of have read The Black Swan before you can really take in Anti-Fragile. Okay, now before I outlay some of these heuristics, let me now just sort of start to try to outline one of the concerns that Nassim Taleb is always sort of thinking about, what his mind is sort of framed around. And it's sort of framed around the statistical issue of the bell curve. So if you have like a bell curve, a normal distribution, okay, of probability. So that means that things in the middle are the most likely to happen. This is the average case. These are like the outliers, the top 10%, the smallest 10%. Okay, so you could imagine the bell curve. This would be something that looked like height of humans, okay? There's never going to be someone who's smaller than, say, a certain size. There's never going to be somebody who's smaller than like 1 foot, okay? Unless you're talking about like little tiny like premature babies or something like that. But okay, get real. Like, okay, we're talking about adults. And there's never going to be anybody who's beyond like 10 feet, okay? So height of humans is a good example where the bell curve, the statistical chance of things happening down here, it actually goes to 0. You're never gonna find a human who's 100 feet tall. It's impossible. And you're never gonna find a human that's like an inch tall if it's an adult. Okay, so this is a good scenario where a normal distribution of probability of events is true, in a sense that the tails, okay, down here, they go to 0. They end. You're never gonna find stuff out here, and you're never gonna find events out here. Now, what Nassim Taleb is usually thinking about and what he's concerned about is not a normal distribution scenario. What he's concerned about is there are other probability distributions that look like this. And these edges are what are called fat tails. Okay, and these are probability distributions where the average outcome looks like this right here, whatever this value is. But it never really goes to 0. That means there's a chance, there's always a small chance that something of gigantic scale can happen. And what Nassim Taleb is worried about is when people confuse this probability distribution with this probability distribution. So let me give you an example that will sort of highlight this confusing these two. Okay, so one, you might think, if you're an investor in the stock market, you might think to yourself, it's highly unlikely that the stock market will crash tomorrow, right? Like, this probability distribution could be the scenario where you're looking at average gains in the stock market. Okay, you might say that this region right here is say, like, maybe plus, I'm not a stock market person, but let's say this is a positive small number where the stock market grew a little bit. This is most likely to happen every day. Okay, and these scenarios here are where the stock market went down a little bit. This is their probability of happening. Here are scenarios where the stock market grew a lot. They're less likely to happen than the stock market just growing a little bit. And down here in the fat tail is the probability of a stock market crash, where everybody loses everything. And so if you're the investor and you say it's highly unlikely that the stock market will crash tomorrow, so today you make risky investments, okay, that might work out in the short term, okay, but that will never work in the long term. And the problem comes with you confusing this distribution with something that looks like this. If you're to make a gamble every single day consecutively for the next 100 years, you can bet for sure that you're going to be gambling in the stock market on the day when the very, very low probability event finally comes around and happens, the catastrophic event. Okay, you're going to be gambling in the stock market on that day if you're investing every day in the risky strategy that you use. And on that day, your investments are going to be killed. Okay, and that comes from you sort of not seeing. You're envisioning that this scenario is true. It's highly unlikely that the stock market will crash tomorrow, which is true. It's highly unlikely. But if you say this to yourself every day for the next 100 years, over 100 years, you're going to be wrong eventually, okay? Because you're thinking that the probability of the stock market crashing is actually 0 when it's not 0. It's a number that's small but greater than 0, okay? And that is where Nassim Taleb has sort of pointed out a lot of flaws in risk assessment, okay? And risk assessment directly pertains to ecological risk assessment and in the release of genetically modified organisms. Because a lot of times we might say, well, the probability of some event happening is very, very low. Therefore, we don't have to worry about it because the probability of it happening is unlikely to happen. It's so unlikely that it's not going to happen. That's sort of a flaw in logic, because you're only temporarily making that statement. You're saying it's highly unlikely that that thing will happen tomorrow or the next day. But you get this sort of flaw in logic pushes you into the best example of this is the turkey problem. And he outlines this in his book, The Black Swan, okay? And this gives you a good example of what a black swan actually is. So the turkey problem is, imagine yourself as a turkey, okay? And every day you are fed in your little cage territory. Every day the farmer comes and he feeds you. So you say to yourself, today was a great day. Tomorrow will be just as good. Tomorrow the farmer is going to feed me. And I can legitimately make that conclusion because for the last 100 days, however long turkeys live, I don't know how long, for the last 100 days, the farmer has consistently 100 out of 100 times come and fed me and given me food and shelter. And so I'm in a great scenario. And so tomorrow the sun will rise and tomorrow I will be okay and the same thing will happen. Do you see how when you make that scenario, when you make that logical conclusion every day consecutively over and over and over, the problem is the turkey's not seeing the scenario where at some point at the end, when the farmer comes to slaughter the turkey, the next day the turkey's head will be cut off and the turkey will be slaughtered and will be dead. So do you see how this is called the turkey problem, and this is sort of emphasizing the logical fallacy, the fallacy in thinking that you get when you say to yourself things like it's highly unlikely that the stock market will crash tomorrow. When you say to yourself true statements that over time become wrong, okay? And this scenario of the turkey problem, the event when the farmer slaughters the turkey, that's called a black swan to Nassim Taleb. So Nassim Taleb defines what's called a black swan event as events that are catastrophic, highly unlikely, so they inherently cannot be predicted. Okay? The turkey can never predict, based on the data that he has, that eventually he's going to be slaughtered. So the black swan event is when he's slaughtered because he cannot predict that. And these are the kind of events that we worry about when we start releasing and genetically engineering the environment in which we live in. There are things that are unlikely to happen that we cannot predict, that will be catastrophic, and that will inevitably happen if we play Russian roulette over and over and over, day after day after day after day, by releasing genetically modified organisms, okay? So these are the things that we worry about, and I hope I haven't botched that explanation of his philosophy too much. But this is sort of the key problem that sort of frames Nassim Taleb's thinking, and then he wraps many of his heuristics around this problem, how to live in environments where these black swans can happen. How can you protect yourself from black swan events that cannot be predicted by their nature? Okay, so in the light of all that I just talked about, my first heuristic is acknowledge the unknown, okay? Any conception that humans have the ability to predict in the future what's going to happen is a fallacy, okay? We are very, very bad at predicting what's going to happen in the future. So acknowledge the unknown. There are scenarios where we can never test enough N, okay? The turkey can never test enough days to predict that he won't, or to predict that he will be slaughtered, okay? The turkey can never predict enough days. We can never say to ourselves, because the sun has risen every day for the last 2, 4, 5, 10 million years, that the sun will rise tomorrow. That's a logical fallacy. We never know that for sure. Okay? And improbable events like the turkey getting slaughtered or the sun not rising, those things happen over long time scales. They do, okay? So with all that in mind, the planet is not a renewable resource. There are scenarios where we could create catastrophic events that will literally destroy our species or destroy the planet. And we want to avoid those scenarios if we can. So to help that, just acknowledge that there are some unknowns that you can never be sure about. And so this should overwhelmingly allow us to think with a little bit of caution when we acknowledge the unknown. Okay, heuristic 2. You want a little bit of stress. We as humans need to sort of refrain from trying to optimize everything from perfection. Okay, there's an interesting book on what's the name of it? The book is Aerosmith by Sinclair Lewis. Okay, it's actually kind of a terrible book. I tried to read it. I only got halfway through it. But okay, in this book, there's a really fascinating scenario, which I still remember to this day. It's about a rising medical doctor, okay, who loves to do research. And in this book, this doctor gets to know this scientist, Professor Gottlieb, really well. And Gottlieb has this great quote. And Aerosmith's goal is essentially like, well, we want to try to get rid of diseases. We want to like engineer scenarios where we can get rid of all diseases. And Professor Gottlieb poses this question, which is, why would we ever want to get rid of all diseases? Because the inevitable result of that would be that our immune systems would collapse. They would not be used to fighting off diseases. We would completely lose our immune systems. And then when the next virus eventually came, we would be wiped out as a species. So you never want to like engineer scenarios where there's no stress at all, because that sort of decrepifies the systems we have evolved, which are able to handle stress. You always want a little bit of stress on the system that's going to be sort of pushing those immunity systems and strengthening them, because you don't want to get into the scenario where you no longer have those systems to fight off diseases. So we want to refrain from the idea, as biotechnologists, a lot of what we do is, how are we going to engineer resistance to a particular virus? Or how are we going to engineer a particular vaccine? Or how are we going to create an organism that doesn't spread a particular disease? But I think we philosophically want to avoid thinking the idea that we want to rid the world of all stress, okay? I think that we don't want to do that as biotechnologists, because that's going to create scenarios where we become extinct as a species. We want a little bit of stress, because stress is sort of an actor that builds robustness, okay? It's a selective system in evolution that builds resistance and ability to survive in stressful scenarios. Okay, so let me give you two examples of this. Let me give you two great examples of this, which we've already covered in the class, one of these, a good example of this, you want a little bit of stress, is actually the refuge strategy. So if you remember from the BT lecture and the insecticide lecture, the refuge strategy is if you're planting a field of crops, okay? And if you're using 90% BT crops, which are going to kill all the insects, you always want to leave a little bit, maybe in a separate field over here, maybe 10% of your crop you want to leave as non-BT, okay? And the reason for this is because this puts a little bit of stress on the situation. You're going to take a little bit of damage when the insects come. But when the insects come, okay, they're going to go, and they're mostly going to survive in the non-BT environment. These are your little insects, okay? Most of them are going to be killed in the BT field, and there's going to be a lot that survive in the non-BT field. So you're going to lose 10% of your crops. That's going to be the stress. But you're willing to take that stress, because then you know in the next generation of these insects, in the next season, these resistant insects that are resistant to the BT are not going to take over the population. Most of what's going to be reproducing are the non-BT resistant insects, okay? So the next generation, you can bet that you do the same thing. You're still going to get 90% of your crops, and you'll lose 10%, okay? This is the refuge strategy. This is a strategy to mitigate insecticide and BT resistance. It mitigates the effects of that because it allows the wild insects to reproduce, okay? And so this is a strategy, a heuristic of this heuristic 2, which is taking a little bit of stress to preserve the overall strength of this system as a whole. Okay, let me pose another scenario, another example of you want a little bit of stress, okay? So imagine two cities, okay, city 1 and city 2, and these are rural cities with lots of farmers. And city 1 is organized of many different farmers with many different plots, and all these farmers have their own strains of wheat, corn, soybeans, everything like that. They got their own strains. They were passed on from their grandparents, which genetically bred them, and they're all different. So this city 1 has lots of genetic, what we would call genetic diversity, meaning there's lots of different alleles. There's lots of different mutations. There's lots of different genetic sequences floating around in the crops of city 1. Okay? Now city 2. City 2 wants to be maximally efficient at producing corn. So city 2 just plants a giant plot of BT corn. Okay? So what's going to happen over a course of 100 years? In the short term, city 2 is going to produce far more product, okay? Because they're not going to get hit by insects as hard. They're not going to get hit by the viruses that these insects spread as hard. City 2, in the short term, is going to produce far more than city 1, okay? But think about the turkey problem. What's going to happen when someday, maybe after the first decade, a new virus emerges, which is spread by insects, okay? And the virus gets spread throughout the entire city 1 and spread throughout the entire city 2's agricultural produce, okay? What's going to happen? Well, in city 1, some of these plots are going to be lost to the virus, okay? Some of these are going to be completely wiped out because their genetics are not evolved properly. They don't have the right alleles to resist against that virus. And in some of the plots, there's going to be maybe like a 50% reduction. There's going to be a tough loss. It's going to be a tough year for those farmers, okay? But in a lot of the plots, they're going to be okay, because in these other plots, they've got genetic diversity. And so in these other plots, there might be genetic strains which are resistant or which have a particular allele which allows them to defend themselves better, okay? The point is that city 1 is non-homogeneous. It's a heterogeneous population of many different not species, but many different strains of these agricultural products. There's a high genetic diversity, okay? So when the catastrophic virus hits, city 1 is going to be okay. They're going to have enough food to survive, and they're going to share amongst themselves, and all the farmers will rebound the next generation. Now ask yourself the question, what's going to happen to city 2? If this one strain of BT corn, which was maximally efficient at production for the first 10 years, if that strain of BT corn is not resistant to this virus, the entire city 2's population of agricultural productivity, of its agricultural products, is going to be completely wiped out. And city 2 is going to starve to death unless it goes to city 1, okay, and asks for food and seeds. Okay, so this leads to the point. This is an example of heuristic 2, which is you want a little bit of stress. Okay, you want a little bit of stress because it's going to help you over the long term survive through the long term. Okay, and point three, as I just illustrated, is you want genetic diversity. You want to maintain the genetic diversity. And the real risk with GMOs is that risk that, say, a Monsanto or a Syngenta or some company in the future invents a particular strain that is a money-making strain, okay? It's a money-maker because it's not susceptible to the certain viruses of the day, and it kills all the insect pests that are biting it today, okay? And so there's going to be a real problem if all the farmers uptake that strain and all the farmers start growing that one particular strain, because in the long term, that's a gigantic loss of genetic diversity, and that's going to come back to bite you over the long term when a catastrophic virus or a catastrophic insect pest comes and hits that land, okay? That's going to be a catastrophic scenario. You want to maintain genetic diversity. That's heuristic 3. Okay, heuristic 4 is a couple things that are very similar that you want to always keep in mind. One of these is called iatrogenics, which is Taleb, again, identifies in some of his books. This is when, if you go to the doctor, okay, and the doctor tells you to take a certain drug to cure problem X, or get certain surgery to cure problem X, okay? But the doctor doesn't tell you that the result of taking that drug or the result of getting that surgery is that there is a 10% cost, a side effect, a 10% probability that certain side effect Y could happen, okay? And that's a real problem if just sitting around with the problem X only has, say, like a 1% chance to really, really hurt you. So if there's only a 1% chance to hurt you, but the strategy for curing problem X has a 10% side effect, a 10% probability that side effect Y could happen, which is just as bad, it's a really bad idea for you to take the treatment strategy, okay? So the idea of iatrogenics is it's this concept where you are trying to solve problem X, but by solving problem X, you just create a worse problem Y, okay? And we need to think about that because a lot of what we're doing with engineering genetically modified organisms is we're trying to treat problem X. And we never bother to consider what is the cost or the side effects of the treatment to cure problem X. It could be the result of a vastly worse problem Y, which we're not even thinking about, okay? So the iatrogenics is the idea, be careful about your treatment of the problem because oftentimes the treatment causes downstream problems which are even worse than the original problem, okay? You need to keep that in mind. And this is very similar to a concept that another sort of pseudo-mathematical philosopher named Deming. So Deming was an MD who was famous for statistics. He was a sort of a father of business statistics. And he was famous for, after World War II, Deming was famous for going to Japan and he would do training sessions with the Japanese corporate leaders, okay? And he would teach them statistics and he would teach them management based on statistics, okay? And Deming has this principle which he calls tampering, okay? Tampering is very similar to the iatrogenics. It's when you try to cure a problem and you create a worse problem Y, okay? And it's about not understanding statistics. So imagine there is... So imagine you are working in a factory and the factory produces widgets, okay? And let's imagine that all of a sudden some machine breaks, okay? The machine breaks and after the machine breaks, the manager says to himself, this is a huge problem that the machine broke. We need to implement a policy such that this machine this pattern doesn't happen again. And what the manager is not realizing is that the machine breaking is going to happen regardless. It's a statistical scenario that eventually machines break, okay? And so the flaw of the manager is in thinking that it's the fault of somehow the workers or it's the fault of the system itself that this machine broke. He's not acknowledging that there's a statistical chance that through random events, machines break down, okay? And so imagine now the scenario where the manager implements a certain policy, okay, where every day you got to go in and you got to do X, Y, Z, and the goal is to prevent future machines from breaking down. But what if the cost of that management policy, the cost of implementing XYZ techniques to prevent machines from breaking is, say, a huge reduction in 20% of productivity, okay? This is what Deming would call like tampering. It's when there's a sort of a normal problem which the manager envisions as this thing that is caused by the system, when in reality it's just a random event. And then they implement management policies to try to prevent this random event from happening, which sort of disrupt the system which was working prior. That's what he would call tampering, okay? So tampering is it's very, very similar to the iatrogenics in the sense that you're trying to deal with problem Y, and so you try to fix it with solution A, and you don't realize that the cost of solution A is much worse than the actual problem. This is tampering. We want to avoid situations where we're in the short term trying to get a little gain and not realizing that we're producing a big cost in the long term. We don't need to solve every problem, and we need to maintain a little bit of stress on the system. Okay, heuristic 5 is slow down, okay? Young people like me, and I know it's a flaw of my thinking, we think that everything needs to be done quickly, everything needs to be done fast, and the faster we're moving, the better we are in the long term, okay? As people who are potentially going to modify the environment, as biotechnologists that are creating and manipulating organisms in nature, we need to slow down a little bit, okay? And the problem is that the scientific system itself, sort of through this uber emphasis of productivity, pushes us to try to go as fast as we can. But in reality, we want to slow down, okay? We want to make subtle changes and really be careful to measure the costs and the effects of those subtle changes before we try to implement something too quickly. We want to slow down. So in general, the more cautious you are about making a decision, you're going to make a better decision. Nietzsche sort of has a concept of the nobility of leisure, okay? People who are able to have the time to think slowly about careful decisions to make the right decision with the right data, that's sort of a characteristic of a noble nature. We want to slow down a little bit and make careful decisions. Okay, heuristic 6 is test small, test local. And you can remember this from the sterile insect technique lecture. When the U.S. Army developed the sterile insect technique for the screwworm, they didn't start releasing it in Florida right away, okay? They didn't start at the tip of Florida right away. What they did, if you remember from the lecture, was if here's Panama, here's South America. In South America, just north of South America, there's a little island right here called Curacao, okay? And before the United States decided to release a whole bunch of, I guess what you would consider to be called manipulated organisms throughout the entire country, airdropping them from planes, they first went to this little island. And the reason they went to an island was if something bad happened on that island, it's not going to escape to the mainland. It's going to stay on that island. It's like Jurassic Park. It's exactly the example of Jurassic Park, okay? They built Jurassic Park on an island because if something happened, then it would be confined to that island, okay? It's the exact same principle here in the sterile insect technique. We first treated it on Curacao, and because it worked in Curacao, it worked really well and there were no bad side effects. So then we went to Florida and we started testing right in the tip of Florida, releasing the sterile screwworms. And then it started working on the southern tip of Florida. And then, only then, we decided, okay, now we're going to really push the screwworm back throughout the entire United States, all the way down to Panama, okay? So this is an example of test small, test local. If you have a new genetically modified organism that you just built, you first want to test it in the lab, and you want to test it locally. Okay, locally means it can't get out, it can't spread. So that means a couple things. It means A, containment. It means it can't get out. So in the lab, when we're making genetically modified insects, we're very careful to keep these behind certain barriers. Like if you make a genetically modified mosquito, it's important that you keep that mosquito behind, say, in an incubator. And then that incubator is stored in a special room that always has a closed door. And on that closed door are flaps and negative air pressure such that the mosquito can never fly out. And even if it flies out, the next room that it goes into is cold, such that if it flew into that third room, if it was able to get out past those first two barriers, it's now in a cold room. And so it would get cold and it would die. And then there's another door. So it's behind another door. It's behind a third door. And then there's a fourth door, which is out into the hallway, okay? And you have special barriers on that fourth door. So this is the idea of we have levels of containment, okay? And the island strategy doing something first on an island is an example of this containment strategy. It's an example of testing local. Everything first needs to be tested local and tested small in a situation where it can't escape and cause vast problems to the entire globe. Okay, my 7th heuristic is that less is more. Simple solutions by their nature are always going to be more attractive. An example I give for this is the scenario again of mosquitoes. Say you want to prevent malaria, okay? And you have two options. One option is to genetically engineer a modified mosquito, which is going to kill malaria in the midgut, okay? And you don't have any idea what the cost of that releasing that genetically modified organism is. You don't know what the downstream side effects are going to be on the malaria parasite or the ecosystem or the ability of that mosquito to spread that disease. Okay, so that's one strategy, genetic modification of a mosquito, okay? That's a very, very complicated strategy, and there's so many variables that you have no idea what the outcome is going to be. The other solution is put screens on your windows. That's going to help malaria just as much, if not more, if not vastly much more, than the genetically modified mosquito, okay? And so let's do simple solutions first. Let's first try to raise the structural integrity of everybody's houses before we're trying to engineer the ecosystem. Let's first make sure everybody has the possibility to sleep underneath a bed net so that they're not being bit by mosquitoes. They don't have open windows. They have a roof over their head. Simple solutions first before we try to vastly engineer the ecosystem to prevent somebody from getting malaria. Less is more. And I would just reiterate that even this scenario one, even if this is the way that you got to go and this is the way that you're doing it, you got to realize that this strategy is a linked chain, okay? And every piece of these chains has to happen for this scenario to work. You say your mosquito, you have to be able to insert the gene. The mosquito has to maintain its fitness, okay? It's got to actually work in the wild. All these things, it's got to be expressed. It's got to have a right promoter. All these things are links in a chain that if any one of these things breaks, if any one of these pieces breaks, the scenario doesn't work. Okay? So you gotta understand that all these genetically modified strategies we're working with are these detailed chains. And if you're not careful, these things are inherently built to break down, because as soon as one of these chains breaks, the whole strategy collapses. So always try to favor a simple solution before you dedicate your life's work to something that's super complicated and relies upon a chain of linkages of end length long. Okay, my point 8, heuristic 8, is we should be thinking about philosophical conservatism. Now, this is going to get me in trouble if people misinterpret this, so I want to be careful, okay? What I'm saying, I'm using the actual definition of conservatism, okay? What conservatism is it's the idea, it's a philosophical principle that you do not try to change systems that have worked for a very, very long time. Because, again, it goes back to iatrogenics. If something is working, why break it? Why change it? Okay? So there's an idea of philosophical conservatism. Don't change things that have worked for a very long time. And this applies to the environment. And it also applies to evolution, okay? Things in nature that have been working for millions of years are time-tested, robust systems that have been exposed to shocks over that time and have survived a long time. So when we start to think in our arrogance that we have a great idea, that we're just going to engineer the ecosystem to be better than it is, and it has survived thus far for millions of years through evolutionary shocks, that is a vast sort of arrogance that's going to lead us towards bad scenarios. We need to be very, very careful when we're trying to change things that have lasted for a long time. We need to sort of be resistant against the idea that we have the knowledge to transform these systems. Okay, and point 9 is skin in the game. And this is a direct quote, direct point from Taleb's book, Skin in the Game. And this is so popular now that I almost feel bad for talking about this, because like everybody is just like going around saying, yeah, skin in the game, skin in the game. So I kind of feel bad talking about it. But I agree with the principle, okay? The principle is that in this case, in our case, what we're thinking about is engineering ecosystems and changing sort of ecological systems. If you're not a person that actually has to live in that ecosystem, you should not be making that decision. So a really important point in sort of the factors of releasing a genetically modified organism, a GMO, or changing the ecosystem in some way, doing some particular treatment, doing some particular test, you need to get community approval of that, okay? So like, for instance, in the Riddle system that we talked about, where Oxitec made the Riddle mosquito, it's important that when they're going to release the sterile insect technique mosquito, the Riddle modified mosquito, that they go to that Brazilian community and that they get approval, because it's not okay for like European English people to be engineering mosquitoes and then being like, well, we're not going to release them in our own community. We're going to go to Brazil, a third world country or whatever, and we're going to release them in this little town. Like, that's not okay, because the Europeans then in that scenario don't have to deal with the ecological consequences if something bad happens, okay? So a skin in the game, the principle is that you shouldn't be proposing these giant changes in systems if you don't have to actually live in that system. You should be the person who maintains like a presence in that community, and you should get community approval before you sort of think in your arrogance that you can go out and do these things. Okay, so these are my guiding heuristics, which I think all biotechnologists should think about, the 9 guiding heuristics that should guide sort of thinking before you decide to make a genetically modified organism, and before you decide to try to change something gigantic. You should think through these things, and they shouldn't make you afraid. I mean, as humans, we have to progress. We have to make progress, and we have to, I think the scientific sort of pursuit is gonna continue. It's a momentum that's not going to stop. These things are going to continue. These genetically modified organisms are going to continue. Eventually, humans have now already been genetically modified with CRISPR, and that's just going to get more. That's going to happen more and more and more. Genetically modified organisms are going to become more robust, more apparent everywhere in our life, in our ecological systems, and in our habitats that we live in. So, with that understood, as future biotechnologists, you should consider these principles before you decide to change something without understanding the cost. I will end there.